Hello everyone, let's talk about the basics of ECG. The ECG, it records the electrical activity that is going on in the heart, and actually it records the currents which are flowing on the surface of the heart. Okay, so in before the SA node it starts uh, firing an impulse, all of the cardiac muscle, it is in a state of resting membrane potential. Okay, so when it is in the resting state, the inside of the cell will be negative. So it will be a state like there will be a positive charge on the outside and a negative charge on the inside. Okay, so if you connect two regions by means of a voltmeter having a negative and the positive electrode, there will be no voltage or no current will be flowing through, because these are having the same potential. There is no change in the potential difference. Now if one part of the heart it gets depolarized, what will happen? So this inside will become positive and the outside will become negative. Okay, when this happens, you will see some deflection in the positive side, because the negativity is coming through the negative electrode and positivity from the positive electrode. So you will see a deflection in the positive direction. Okay, now as this wave of depolarization moves further, more and more regions, they become inside positive and outside negative. So a state like this happens sometime. Now when this state is there, you can say that there is a maximum number of negative charge and maximum number of positive charge. So the current flowing through this will be the maximum. So the deflection which you see will be in the maximum positive direction. Okay, and after this, what will happen? If the depolarization goes on further, this also becomes negative, this region also becomes negative, and it proceeds further till the end. What will happen? All of the myocardium now is in the state of depolarization. So when it is depolarized, these two ends again are having the same potential, though it is on the opposite side, that is negative. So again you will see no deflection in this case, so it will be like this. Okay, so when it starts, whenever the depolarization starts, you can see a small upstroke, and as it becomes midway somewhere, you will see the peak, and then it will go back to the same state as it was before. Okay, now when repolarization occurs, then the again process will be repeated, but in an opposite direction, because generally the tissue which depolarizes first, it also repolarizes first. So the changes will start occurring in this direction. So you will have positivity here to begin with and negativity inside. So now what you will see, there will be a deflection in the negative direction, because the negative terminal is on the positive side now. So you will have a deflection in a different direction. So then again, as it proceeds further, more and more, then the deflection at this point will be maximum towards the negative side. Then going further, when the cells come back to the resting membrane potential, once again you can see there will be no deflection. So there will be again onto the middle path. It stays in between because there is no potential difference on these two sides. Okay, this is how depolarization and repolarization occur and how they are recorded by means of a voltmeter or ECG leads. Now this, the heart it lies in an oblique direction in the chest, in the body. So the heart, it lies somewhat like this. Okay, to one side. So whenever, and the SA node is present somewhere over here. So whenever a wave starts, it starts in the SA node and then it goes on depolarizing the atrial muscle. Okay, then it passes through the AV node into the bundle of his, and then it goes further and into the Purkinje fibers and the whole of the ventricular muscle. So you can see that generally this wave goes in this direction. The depolarization occurs in a direction which is downwards and towards the left. Okay, so this can be given a direction which would be somewhat like this. In case of atria, it would be somewhat like this, and in case of ventricles, it would be somewhat like this, along the length of more towards the left side. So this arrow that I have made, these arrows, they have got some important things. One important thing is they have got a magnitude. Magnitude means how large that deflection was, or how large the potential difference was. So that is one thing, the magnitude. And second thing is it has got a fixed direction. That's why it is called as a vector. It has got two important properties. One is it has got a magnitude which can be given by the length of the arrow, and then it has got a direction which can be given by the direction of the arrow. Okay, so these vectors are actually recorded on the ECG by means of leads. Okay, and the vectors, they can be projected onto various axes. So if I have got a horizontal axis and a vertical axis around this, I can give projections of this vector on these two axes by just drawing perpendicular lines from the axis to this arrow. So this much would be the projection of this vector along the x-axis, and this much would be the projection of this vector along the y-axis. Okay, but when we are using the ECG, we do not have perfect horizontal and vertical axes. We have a triaxial system of vectors. Okay, and that depends upon the placement of the various electrodes on the body, and how these electrodes are placed is given by an Enthoven's triangle, which is given by an equilateral triangle made around the heart, somewhat like this. Okay, and these three corners of this triangle, they represent, this is the right arm, this is the left arm, and this is the left leg. Okay, and these three lines, or these three sides of the equilateral triangle, are the three bipolar limb leads. Okay, so this is given as the lead 1, this is lead 2, this is lead 2, and this is lead 3. Okay, and what is the direction of these? This is the negative electrode for the lead 1, and this is the positive end for lead 1. Okay, for lead 2, this is the negative end and this is the positive end. Okay, and for lead 3, this is the negative end and this is the positive end. Okay, so now these three leads, how they are arranged with respect to each other is, one is this, lead 1, Then at 60 degrees to lead 1, you have got lead 2. And at 60 degrees to lead 2, you have got lead 3. So this is lead 1, this is lead 2, and this is lead 3. Okay, so the angles they have in between them, lead 1 and lead 2 are at an angle of 60 degrees to each other. Okay, and lead 2 and lead 3 again are at an angle of 60 degrees to each other. The positive direction being here, the positive vector here, then here and here.